Hello, I'm Jose.
Hello, I'm Isaac.
Hello, I'm Brian.
And I'm Alexis.
And this is Biotechnology Knocking Out Genes. This is our overview slide. Let me give you guys a little bit of background on Arabidopsis thaliana. It is a member of the mustard family. It has a 6 to 8 week generation time. It is self-pollinating. There are about 200 visible phenotypic markers, and all 25,500 genes have been sequenced. Here's an article that I found which was written by David W. Mankey, Michael Cherry, Caroline Dean, Stephen D. Rounsley, and Martine Korneef. And it's called Arabidopsis thaliana, Model Plant for Genome Analyst. And he talks a little bit about the biology of Arabidopsis thaliana. He mentions that it is common in Europe, Asia, and North America. Its entire life cycle is complete within 6 weeks. He also talks about the Arabidopsis community, which is a diverse group of scientists from 30 different countries, all devoted for studying the plant's genotype. He also mentions that in each university there's at least one student in the science department that is at all times studying the plant. He also mentions the vision for the future, which in the short term, he is hoping that we complete the genomic sequence and study the knockouts of major genes. In the long term, he is determined it's to determine the function of key gene products and to study evolutionary relationship between eukaryotic organism and evolution of common cellular and developmental pathways. Another article that I found really interesting was about an experiment conducted by Kosuke Hanada, Takashi Kuromori, Fumiyoshi Mionga, Tetsuro Toyota, Wenxiong Li, and Kazuo Shinozaki, and they talk about duplicate genes in Arabidopsis thaliana, which is something that does happen in many organisms, and it's something that is very important to this experiment because we are studying the phenotype. So he talks about that due to the vast majority of genes present, there are bound to be many duplicate genes, and this may result in there not being a change in the phenotype. They came to a conclusion that there are only changes in the phenotype when there are multiple duplicate genes knocked out. Our question and hypothesis, what we came up with is it possible to use the same techniques used to knock out genes in Arabidopsis thaliana to knock out cancerous genes in human bodies as a treatment option? I mean, could you imagine that? That'd be amazing. Somebody going and getting a cancer treatment where all they have to do is inject you with certain genes that'll knock out the cancerous genes, which, by the way, are always multiplying and replicating. Our hypothesis is UV exposure, high salinity, and drought conditions will lead to changes in the plant's phenotype. Our null hypothesis would be that there is no difference between the wild type seed grown plants and the mutant type seed grown plants. This is not the case, as in most of the scenarios there's at least one difference between the two plants, as you'll see later in our trials. Our variables are our control variable for control group is the wild type seed. Our control variable for experimental group is the mutant seed. Our independent variable is the mutant seeds and environmental conditions. Our dependent variable are changes that we view in the plant's phenotype due to the environmental conditions. And I'm handing it over to Isaac.
Hello, guys. I'm gonna talk about the materials we needed. So this experiment is obviously a virtual experiment. So all these materials are that we saw during like the trials and all that. So first of all, we have our wild type and mutant Arabidopsis seeds. In conjunction with that, we had our pods, the water that we use in the enclosures that kept everything together. And then we had experimental conditions, which were the UV high exposure UV light, the high salinity, and the drought conditions. And then to observe all those things, we needed the magnifying glass. So for the experimental process, First, we inserted wild type seeds and put them in the control plots, and then we did the same with the mutant seeds. We put them in the experimental group plots. And then after that, we were allowed to pick an environmental condition, such as the drought conditions, the UV exposure, and then once we did that, we set them to grow, and then we used the magnifying glass to see any changes in the phenotype. And from there, we used the lab manual to see what these changes meant and like what they did in terms of function. And then at the very end of that, each experiment, we cleaned the pots and we did the process using different environmental conditions until we got all our data. Right here, we have trials 1 and 3. You can see in our graph, we structured it so we can see every single type of mutation that could occur. And for the first one, we obviously see high salinity showed us darker color and they were clumped together, which could be attributed to like the high salinity. So they need that being clumped together to like get as much water to photosynthesize. And then we see plants 2 and trials 1 and 2. We saw UV exposure had less stems and leaves, and then they're lighter in color. Same thing with trial 3. In trials 4 through 6, we saw varying degrees of mutations across all factors. Some were darker, some were lighter, some had less, some had more leaves. But something that I took note of was UV exposure, high salinity, and drought conditions. We saw kind of like small little pores that the leaves would grow. And this could be attributed to like cactus. So that's like parallels the desert in that way under these environmental conditions. Trial 7 through 9, we see the same thing, but actually in trial 8 and then 10, as you guys will see in the next slide, we saw no mutations occurred, which just showcases that just because like we do all these environmental conditions, that won't always change like the phenotype in knocking out genes. And then trial 10 shows the same thing. And then here's our results graph. So on the vertical axis, we have the number of mutations in a plant. The most mutations we observed during all the environmental conditions throughout the 10 trials was the high salinity that always showed us like the 3 varying degrees of mutations. And then throughout all that, the second one was UV exposure followed by drought conditions. And then we can also see optimum growth conditions. There's going to be no mutations due to no changes in the phenotype. Now I'm going to take it on to Alexis.
Okay, so basically for our next slide, we have to decide whether our results agree or disagree with the hypothesis that we came up with. And basically our results did agree with the hypothesis, but only to a certain extent. And some of the differences between the UV exposure, high salinity, and drought conditions in comparison to the optimum growing conditions were substantial in appearance and in function. And in the way that it might not agree is that out of the 10 trials that we performed, only in 8 of them we did see results, which was a difference between the wild type seed and the mutant seed. And lastly, for our conclusion, the results of our experiment showcase that some environmental conditions do affect the genotype more than others, such as high salinity brought the most mutations, which was followed by UV exposure and drought conditions. And in this, we can deduce that knocking out genes is still in a trial and error era, which gives way more in-depth research being done in that specific field. And This experiment gives way to the future in knocking out genes for future medical and experimental procedures.
So, the broader impacts on future experiments. We had some weaknesses and some strengths. Some of the weaknesses were it was based off of a computer, it was randomized, so it was we didn't go through trial and error, and we didn't observe it ourselves. Some of the strengths, though, it was the cost of the experiment. We really didn't have to buy anything for it. We just had everything already prepared for us, and we didn't have to wait for the time period for the plants to grow and actually make it ourselves. The broader impacts on humanity. It is possible to cure cancer, or it would be possible to cure cancer throughout knocking out the genes, but there's a possibility of always for the genes to duplicate. It is also a risk knocking out genes since it can go sideways during the DNA sequence. But with our research, it can help the Arabidopsis community with future research by helping them discover new mutations to help out the new plants survive. And in further research or experimenting, it can be difficult in humans due to the fact that our immune system has to get used to it. And by knocking out the genes, it can lead for cancer to find another way to duplicate. And then these are our references. And thank you.